Welcome, everyone. Thank you for joining us for today's webinar, Studies on Malignancies with a Novel Proliferation Biomarker. It is presented by Martin Shaw, the Business Development Manager at AroCell. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Martin Shaw. I will now turn the presentation over to him.
Okay, thank you for joining me, ladies and gentlemen. Good morning or afternoon, depending on where you are in the world. I'm going to make a presentation on a novel proliferation biomarker and its use in malignancies. In particular, I'm going to concentrate on the proliferation biomarker thymidine kinase 1. It's quite a long presentation, so I'm going to break it up into sections, approximately as shown here. First, a bit about, in general, about tumor biomarkers, then thymidine kinase as a tumor biomarker, its role in hematological malignancies, in solid tumors, in drug discovery, a bit on the assay, and then a summary and discussion. It's a long presentation, so I'll be repeating parts of it to make it easy for you to follow, I hope, and also for those that are only interested in certain sections of it. So what is a tumor biomarker? It's any substance that's associated with the presence of a tumor. There's only three sorts I'm going to talk about now, basically genetic cell surface markers, serum protein markers, and then proliferation biomarkers. I know there are other biomarkers such as metabolomics, but I'm not going to discuss them today. Talking about genetic or immunohistological biomarkers, and there's a lot of these, HER2, PD-L1, AR7, and others. They aid in the choice of therapy, but they do not indicate therapy success, and they're not indicators of tumor growth. There's also serum protein biomarkers, and these mostly indicate tumor mass. And there's many of these, again, CEA, PSA, and many more. But there are very few serum biomarkers of cell proliferation. And many serum biomarkers are inadequate, particularly for advanced disease. And this has been stated for two of the most important cancer types. For example, if for PSA in prostate cancer, you must be stressed that PSA level is not an absolute marker of escape. And you can have clinical progression with normal PSA. In breast cancer, it's been stated not one of the circulating markers has shown clinical utility to support its use as an independent predictive biomarker. And predictive is something I'm going to come back to. So our hypothesis is that most current tumor biomarkers measure what has happened, the past rate of change, such as doubling time, the current situation, such as tumor mass or white blood cell numbers, but what really matters is tumor growth. And proliferation biomarkers can help to show this. And they can aid in choice of therapy. For example, if they're low, watchful waiting, or less aggressive therapy. And if they're high, more aggressive therapy. And they can provide a measure of tumor growth, and they can aid in stratifying patients and subjects in studies. And one reason to only treat those tumors which are growing is that anti-tumor drugs are toxic, they have side effects, they're expensive, and they drive therapy resistance. So going on to talk a bit more about proliferation biomarkers. Most of them at the moment are immunohistological biomarkers. The gold standard is Ki-67, a protein expressed in cells during the growth cycle. PCNA, which I'm not going to talk about, and then TK1 or thymidine kinase 1, which I'm going to talk about mostly for the rest of this presentation. Thymidine kinase 1 is a key enzyme in DNA production, and it increases during the S phase when DNA synthesis occurs. So the percentage of cell staining is the proliferation index, and it provides a measure of the rate of tumor growth. But these are immunohistological procedures. They require biopsy, they're invasive, they're labor intensive. They only show the status of a small part of one tumor. If there's metastasis, what's going on there? And they're very low reproducibility because they're subjective. So there's a need for a biomarker that indicates the ongoing body activity of the malignant disease, that is applicable to a standard serum sample, and that can be determined with a routine analytical method. And I'm going to present to you that serum thymidine kinase 1 has the potential to be such a marker. A little bit about what serum thymidine kinase is. Excuse me. Serum thymidine kinase has been an established proliferation biomarker mainly for hematological malignancies for 35 years. It's been studied in many types of solid tumor, and when TK1 is used in immunohistological staining, it is highly associated with the gold standard proliferation biomarker Ki67. What does it do? It couples phosphate to thymidine as a key step in DNA production, and this coupling of phosphate to thymidine forms the basis of the enzyme activity assays for thymidine kinase 1. As I mentioned, in solid tumors, it's been found in many types, probably it's expressed in all of them. Going on to look at the application of TK1, I'm going to talk first about hematological malignancies. And most of increased levels of serum TK1 activity are found in most forms of leukemia. And it's been used for a long time in many ways. For example, it can be a discriminator of disease stage, provides prognostic information, can aid in monitoring therapy response or relapse, and in disease progression. Looking at clinical staging, this is a study on non-Hodgkin's lymphoma, and as you can see, as the disease progresses, the serum TK1 activity increases. This is a study on chronic lymphocytic leukemia, and again, you can see as the disease progresses, the RAI stage increases, the serum level of TK1 increases, and interestingly, when an elevated TK1 is found in subjects who have an apparently indolent disease not progressing, they have an increased risk of disease progression. It can be used to make prognoses. This is in chronic lymphocytic leukemia, and you can see in those which have a low TK1 level, less than 7 units a liter, the 50% survival time is about 6 years. But when they have a high level, the 50% survival is only about a year. So it's a very strong prognostic factor. Similarly, in non-Hodgkin's lymphoma, in those who have a low level, you can see that 50% of those survive around, again, about 6 or 7 years, whereas those who have a high level survive, again, only about a year. Excuse me. Note that the level of TK1 is different. This is because of the different cell types involved and the different patient populations. To be used to look at disease progression. Here's a study on acute myelolytic leukemia. And the top 2 subjects, which are stable course, you can see that TK1 level stays low. And in the bottom 2, which led to death, you can see that prior to death, there's a rapid increase in the serum TK1 levels. It can be used profitably together with other biomarkers. And serum TK1 activity seem to reflect a combination of tumor mass and proliferation rate. One could perhaps oversimplifying say it gives an indication of the number of cells which are growing. It correlates with the proliferation index, as I mentioned, the gold standard for assessing tumor aggressivity. It correlates with the peripheral blood lymphocyte count, so it correlates with tumor mass. So why has it not been widely used? Well, one problem is the assay. Intracellular active TK1 is a tetramer, but serum TK1 exists as complexes of different sizes and enzyme activities. Some sera contain TK1 inhibitors, and enzyme activity assays may underestimate serum TK1. The gold standard assays for TK1 utilize radioactive tracers. Commercial assays may require specialized equipment. And we're going to put to you later that immunoassays can solve these problems. So I'm going to first talk very briefly about the immunoassay of thymidine kinase 1, particularly with the AroCell TK210 ELISA. In serum, TK1 forms complexes of many molecular weights and enzyme activities, but they all express the TK210 antigen. And as you can see, when you plot the enzyme activity against the molecular weight and at the same time do Western blotting on the different fractions, you can see that where there is enzyme activity, there is the presence of TK1, and you can see it by looking at the TK210 antigen, an antigen specific for the TK1. The AroCell TK21 ELISA, it uses monoclonal antibodies against the unique amino acid sequence, the TK210 antigen. And importantly, a unique pre-assay treatment releases TK1 from the serum TK1 complexes and exposes the TK210 antigenic sites. When you look at the diagnostic power of the enzyme activity assay and the TK210 ELISA, they give very similar data. And this is good because it means that you can use the ELISA, a superior analytical method, and build on the historic data. We've done some early studies on using the TK210 ELISA in hematological cancers, and you can see that in healthy subjects, it's low. In subjects prior to treatment, it's elevated. During treatment, it's up and down, depending on how the state of the patient. And then after treatment in remission, it goes down to approximately normal levels. So talking about TK210 ELISA in hematological malignancies, it's a biomarker of cell proliferation. Serum levels reflect disease activity. TK1 can aid in patient subject classification. TK1 can be used for prognosis. TK1 can be used for monitoring disease progression. It adds information to studies performed with other biomarkers, and it can be used in many malignancies. It gives a good agreement with clinical results based on TK1 enzyme activity. It gives the convenience of microtiter ELISA. It's an open system, and it's CE marked. So that's a bit about the almost historical data on the use of TK1 to study hematological malignancies. But now with the improved technology, one can use it more to look at solid tumors. As I mentioned earlier, TK1 is expressed immunohistologically in solid tumors. Many have been shown, probably all, but it's not been widely studied in solid tumors. And the reason this goes back to the fact that in serum, TK1 circulates as complexes with different molecular weights and different enzyme activities, but they all express the TK210 antigen. And more than that, many of the TK1 aggregates in serum from solid tumor subjects are enzymatically inactive, but they all express the TK210 antigen. This can be shown here in a subject with prostate cancer. The open circles show the distribution of enzyme activity in blood donors. The black diamonds show the distribution in a subject with prostate cancer. And underneath, you have the Western blotting for each of these fragments. And I want to draw your attention to fractions 14 onwards, where there's a strong immunological expression of TK1, but no enzyme activity. So in subjects with solid tumors, the ELISA assay is more sensitive because it picks up these TK1 forms which lack or have low enzyme activity. And as I mentioned, the ELISA, AroCell TK210 ELISA, has a unique pre-assay treatment that breaks up the serum complexes and exposes the TK210 antigen. And here you can see the effect of this in subjects with breast cancer. On the TK210 antigen, the TK210 concentration, the area under the curve is 0.9, but the TK1 activity, it's 0.79, so you have a better diagnostic power. But more importantly, if you look at the sensitivity, for the TK210 concentration is 0.5, and for the TK1 activity is 0.26, both with similar specificity. So the ELISA is about twice as sensitive for measuring TK1 in subjects with breast cancer. As I said, it can be combined profitably with other tumor markers. Here's a study where they looked at CA15-3 in breast cancer, and you can see the blue line, which is CA15-3, and you can see the red line, which is the TK210, and then if you combine them, you get the black line, and where you have even better performance. So this is a good point, because when you combine the mass, it's an indication of mass, such as CA15-3, with an indication of proliferation, such as TK1, you get a better result. This has been shown similarly in prostate cancer. You have comparison with pro-PSA. The red line is the pro-PSA. The bluish line is the TK210, very similar. But if you combine them, you get a bit better result there. So you can say that in separating subjects with potential prostate cancer from subjects which are found to have prostate cancer, TK210 is about as good as the new prostate biomarker pro-PSA, but combined with pro-PSA is even better. So looking at solid tumors, the conclusions: the AroCell TK210 ELISA detects TK1 forms missed by enzymatic assays. The TK210 ELISA gives better discrimination between healthy controls and subjects with solid tumors. Can be used to study many types of tumor. Adds information to studies performed with other biomarkers, and it's CE marked. So that's a bit of the use of the TK1 immunoassay and TK1 activity assays in malignancies. I'm now going to talk about it a bit in drug discovery. A key step in all drug development is this evaluation of the product in vitro. And the assay of TK1 with the AroCell TK210 ELISA kit offers the potential to study anti-proliferation effects in vitro. There are many biomarkers of cell death in vitro. The TK1 assay is an indication of cell growth in vitro. Here's like proof of principle. Here you're looking at the concentration detected for TK210 compared with the number of cells present, and you can see there's a pretty good correlation. Here's a study where a leukemia cell line has been studied in vitro, where the cells have been exposed to the anti-cancer drug doxorubicin. The blue line is the intracellular content of TK210, TK1, and the red line is the concentration of TK1 in the supernatant. As the level of drug administered increases, the first thing you see is that the level of TK1 intracellularly increases, and this is due to the fact that doxorubicin attacks DNA, and the cell is trying to grow more DNA, so you get an upregulation of TK1. As the dose of drug increases, the intracellular concentration of TK1 falls, and you start to see the appearance of TK1 in the supernatant as the cells die. And this is very interesting because in cell culture, in clinical trials, when you use thymidine-based scanning agents to do positron emission, you can see at the initiation of therapy, the tumors glow. You get what's called a flare, which disappears as the drug becomes more effective. So what you're seeing in the microtiter plate is similar to what can be seen in clinic. So looking at TK1 in the microtiter plate, it shows it's potentially a translational biomarker. We're also seeing now that TK1 seems to be a very good complementary diagnostic to CDK4/6 inhibitors. These prevent cells entering the S1 phase, and TK1 is present in the S1 phase. So the effect of CDK4/6 inhibitors can potentially be seen very clearly by measuring changes in TK1. Similarly, hormonal therapies seem to do the same thing. Following studies in microcytoplate, the next step is to look at xenograft studies. And here's an advantage of ELISA compared to enzyme activity assays, because mouse and human TK1 are immunologically distinct. So you could potentially get improved monitoring of xenografts in mice. We are performing studies on this, and if you've got any studies in this field, we'd be very pleased to hear from you. So I've now gone through showing that TK1 ELISA can be valuable in clinical studies and in drug discovery. I'm now going on a little bit on the assay method. And I'm going to divide this part up into some small parts: the analytical challenges, the assay itself, performance data, the handling of TK1, further information and discussion. So as I mentioned twice already, molecular weight of TK1 complexes and their specific activities differ between normal and tumor subjects. And some TK1 aggregates from serum for solid tumors are enzymatically inactive. Here you can see in hematology subjects where there is pretty much the expression of TK210 where there is enzyme activity. But you can see here that in solid tumors, a lot of the TK1 is not enzymatically active, but it still expresses the TK210 antigen, and so can be measured by the AroCell TK210 ELISA. The pre-treatment of the samples releases the TK1 tetramers from the line weight complexes and exposes the TK210 epitope, so you get greater sensitivity for solid tumors and more consistent results between subjects and types of malignancy. Describing the kit very quickly, it's a microcytoplate sandwich ELISA. It uses monoclonal antibodies against the epitope TK210. And most importantly, a unique pre-assay treatment releases TK1 from the serum complexes and exposes the antigen sites. The method is very simple. As I mentioned, the pre-treatment of the sample. You add it to a microcytoplate coated with anti-TK210. You then add a second anti-TK210 antibody labeled with biotin, and the color is developed using ultra-rich peroxidase labeled with streptavidin. It's got a wide assay range. It's got very sensitive. It's got linear dilution. It's very precise, and it's got long shelf life, so you can keep it on stock until the samples arrive. TK1 is quite an easy protein to work with. It's set at room temperature while you're working with it. It could be kept at 4 degrees for a short time, so it could be shipped. And then for long-term storage, it can be kept frozen, so you can do retrospective studies or prognostic studies. As I mentioned, in hematological malignancies, it gives quite similar results to the traditional TK1 enzyme activity assays, so you can build on traditional data. In solid tumors, in this example for breast cancer, it's much more sensitive for TK1 than the enzyme activity assays. It can be profitably combined with other tumor biomarkers. Publications are coming out on its application, such as these examples. So, getting to the conclusion, TK1 is a biomarker of cell proliferation. It's a forward-looking biomarker. Serum levels reflect disease activity, patient-subject classification. They can be used for making prognosis and for monitoring disease progression. TK1 has different forms in solid and hematological malignancies. Enzyme activity assays underestimate TK1 in solid tumors. And AroCell TK210 ELISA measures all forms of serum TK1. So you get good agreement with clinical results based on TK1 enzyme activity assays in the hematological malignancies. Greater sensitivity for solid tumors greater sensitivity for solid tumors and more comparable results between types of malignancy. It's a standard ELISA procedure. It's applicable to in vitro models. It's an open system and it's CE marked. So thank you for listening to me, ladies and gentlemen. I've been presenting the AroCell TK210 ELISA, a novel biomarker of cell proliferation, and as we say, it measures what really matters. It expands and simplifies the study of cellular proliferation and provides new opportunities for cancer research. And I look forward to seeing your questions, and I'll answer them as best I can. Thank you for your attention.
Thank you, Martin, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 27th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.